I would like to introduce to you our speaker today, a dear friend, dear colleague, an amazing scientist, Ana Fernandez-Sesma, who I am co-hosting with Matt Dougherty today. So Ana received her Master in Science degree in Biological Sciences from the University of Salamanca, Spain. I understand she's originally from Salamanca, but I'm not sure. She then received her master degree and PhD degree in biomedical sciences from Mount Sinai Graduate School in New York, and then performed about 6-year postdoctoral training working on dendritic cells with Dr. Thomas Moran, also at Mount Sinai School of Medicine. And in 2005, she was named assistant professor at the Department of Microbiology at the Mount Sinai School of Medicine in New York. And then she she moved into the tenure track and quickly moved through the ranks. And today she is professor with tenure in the same department, the Department of Microbiology at Mount Sinai School of Medicine. So Anna's main research interest is in the study of innate immune evasion during infections with human viruses that are of health interest, such as dengue virus, influenza virus, and other important human pathogens such as HIV and also SARS coronaviruses, but she is not talking much about that today. But I think that I could be biased, but I think we all agree that viral immunology and research into the virus and host interactions are a significant and now a very timely topic. So the main focus of Anna's laboratory is on dengue virus, and I think she will focus the talk on that today. And she has developed and optimized a human primary cell system in which she can study the interaction of dengue virus with human primary immune cells, such as monocytes, dendritic cells, and macrophages, and has recently developed a human tonsil system to study dengue virus infection. And so using these primary human cells, her group has made a number of significant discoveries for the field. And among these discoveries, I would like to highlight 2 discoveries from Anna's team today. The first one is the identification of an interferon antagonist in dengue virus, particularly the protease NS2B3, which is able to cleavage a sting to suppress type 1 interferon in infected cells. And subsequently, another discovery that is the demonstration that dengue virus can degrade the DNA sensor SIGAs in the infected cells. And so I think that these discoveries really illustrate the importance and significance of Anna's work and the type of research she does, because both together they help establish a new and important paradigm in the field, showing that even during an infection with an RNA viruses like dengue virus, which is an RNA virus, the DNA innate sensors and the like SIGAs and the adapter sting can be activated via the leakage of mitochondrial DNA. And Anna's work consistently showed that these RNA viruses have evolved means to counteract or evade DNA sensors. And again, I think that this is an important paradigm that Anna helped establish with her research. So not surprisingly, Anna's work has been published in prestigious journals of high impact in her field. And she has also been tremendously successful at securing external funding for her laboratory, but not only for her laboratory, but also for multiple laboratories by leading a highly competitive multi-PI grant application that supports collaborative research in dengue from many groups across the country. So a testimony of that success in securing fund and also a reflect of her research. is that Anna has been ranked among the top 5 female NIH-funded investigators in microbiology in the US for at least 3 consecutive years. So I think this was between 2015 and 18, if I remember correctly. So I could continue providing details of Anna's contributions as editorial board member in prestigious journals, grant reviewers for NIH, and also international grant study sections as a leader in graduate education. But I think time is precious and we all want to hear from Anna. So I will wrap up there and thank you, Anna, very deeply for accepting the invitation to present today, and also reiterate that this is a real gift to be opening this seminar series with you today. Thank you for accepting.
Thank you, Alina. You already gave my seminar with the introduction.
I try to be brief. Sorry if I extended too
No, it's
much.
okay.
But
It's good. It's good. So I'm gonna try to share the screen, see if I can.
Perfect. Yes.
If it works
And while you share the screen, I will remind everyone that Anna has accepted to record this seminar, and it will be available online at the program of immunology. And if you have questions, you can send the questions via chat, and then Mad at the end will ask you to unmute or will read the questions, and he will be handling the questions at the end. So thank you, Anna.
Thank you. Thank you again. Can you see my screen?
We can see it perfectly well, yes.
Okay, so these are my views from my office. So I am in New York, in Mount Sinai. As you know, as you heard, I spent my whole career in Mount Sinai, so there must be something I like about this place. But anyway, I just wanted to give a kind of a trajectory of my lab. So I told Elena I wasn't going to talk about coronavirus. I will mention coronavirus, but I think there is this coronavirus fatigue on everybody. So I rather talk about like a trajectory of the lab, how we went from molecular pathways in which we were looking of how viruses manipulate innate immune pathways to really try to validate everything we found in the lab using primary cells and also human samples. So hopefully that will give you a much more complete story. So I'm going to talk about dengue because it's a virus that we find very interesting, and also because it's a virus that likes to infect immune cells. So the disease is really an immune pathology. So dengue is transmitted by mosquitoes, so in particular Aedes aegypti and Aedes albopictus, and they are really in tropical and subtropical areas of the world. So that's why 3.5 billion people are at risk of infection every year. So this is a virus that causes tremendous morbidity. Yeah, but it's not a very lethal virus in general. Only maybe 10% of people might develop a severe manifestation of dengue, which is dengue hemorrhagic fever. So as you probably are hearing now a lot with coronaviruses, it's not that uncommon that viruses will have like 80% of people with asymptomatic infections. But nevertheless, if you have a massive amount of people at risk, there is a big morbidity and also higher mortality in the world. So dengue is a big problem in the world. And now with global warming and other changes, it's expanding. The mosquito area is expanding. So this is of high importance. So dengue started as a virus between primates and mosquitoes, and it was a cycle called sylvatic dengue, a zootic cycle. But at some point, it jumped into humans, and it actually established its own cycle between humans and mosquitoes. And one thing that is interesting is that there is really almost no reported cases. There are almost no reported cases of human-to-human transmission of dengue. So there is a need for this vector, mosquito vector, to transmit the virus or for efficiently transmit the virus. So what that is quite intriguing because you need to take into consideration the biology of the vector and the influence of the vector when you're studying dengue disease. But also something that has been challenging to look at dengue virus infections is that there is really no good animal model for dengue, meaning that there is no immune competent small animal model to study dengue. So that has really forced the field to use immune compromised mouse models or other animal models that have made it kind of is an important tool for the field because have made significant contributions for the virology and looking at viremia and how the virus actually replicates. But in my area of research, I'm really interested in how the viruses interact with innate immunity and how we mount an immune response for this virus, it has been like difficult to use the existing animal models. So it's a virus that is a flavivirus, so it belongs to the family of Flaviviridae, and this is just the genetic scheme of the virus. So the virus is a positive strand RNA virus, which means the genomic RNA is a messenger RNA. So the first thing that happens in the cell is that the genomic RNA will be translated into a polyprotein, and it encodes one single long polyprotein with three structural proteins, which are the envelope, preem, and capsid, which are the ones that are visible for the immune system when the virus infects the host. And these are like against which we mount most of the antibody responses. Um, but there are also 7 structural proteins, which are my favorite proteins in this virus and every other virus, because they're really instrumental for the virus, not only to replicate, translate, and transcribe and do all the functions that a virus has to do in the cell, but it also contains molecules or proteins that are important for evasion of innate immune responses. So most of the non-structural proteins have an important role in actually antagonizing the innate immune system. So this is how the polyprotein of dengue is made. And this is a virus that really loves the ER membrane. So the polyprotein wraps itself around the ER membrane. And there is this viral protease that also Lina referred to, the NS2B3. So it's composed by 2 different proteins of the virus. And the thing that happens initially is that it cleaves itself from this polyprotein. So the protease has it can cleave itself from the polyprotein, and then also mediate the cleavage of the proteins that are in the cytoplasmic side of the cell, of the membrane. But then there is also a contribution of phosphoglycinases inside the ER that will mediate the cleavage of all the other proteins that are in the luminal part of the ER, as well as furin that is important for the maturation of the viral particle in the Golgi when it's exiting the cell. And also an unknown protease that mediates the NS1. So this is just a coordinated participation of viral as well as host proteases in order to release the individual proteins of the virus. And I'm just listing here several of the functions that we know of all these different proteins of the virus, but I don't really pretend that people are going to remember or like memorize this. The only thing I wanted to really emphasize here, first of all, my favorite protein, which is the protease, not only has a function in the cell cycle, which is in the viral cycle, which is to cleave the polyproteins, is also an important interferon antagonist, both components of the protease, but also to highlight that this virus has at least 4 proteins, if not more, that are really involved in antagonizing the type 1 interferon system, which means that for RNA viruses and for most viruses, if they don't really dominate or actually inhibit the type 1 interferon system, they will not be able to establish infection. That tells you that there is a significant amount of energy and real estate that viruses have to actually invest in order to have these functions. So that also highlights the multifunctionality of the viral proteins. They really have to serve many different functions, and also the combination of proteins might create a new function for the protein. So the way we see the infection and the way it happens is that the virus will be deposited in the skin by the mosquito bite, and there are dendritic cells, Langerhans cells will actually pick up the virus and will be able to get activated and migrate to the lymph nodes, where they can do an amplification, where there is an amplification of the virus in susceptible cells. So this virus really likes myeloid cells. And then eventually it will reach the bloodstream again, where it can cause viremia, and it can be picked up by another mosquito, so how as the virus transmits. So in the lab, and particularly at the beginning, we use monocyte-derived dendritic cells, which are convenient for us to obtain from healthy donors, and we can actually differentiate them in vitro. And then we can really use them because they are infectable by dengue virus, and we use them, and we believe they might represent different stages of dengue infection. So you probably, for this audience, is not, I don't have to really like dwell very much into this, but one of the things that what we think is important to look at the dendritic cells, in particular in the case of dengue, but in general, is that because they really link this innate and adaptive immune responses, and if they encounter virus or they are infected by virus, like is the case for dengue, they will go through this process of maturation or activation, and they start secreting cytokines, chemokines, including type 1 interferons, and also that is what is the hallmark of innate immunity. And then they also change morphology and they're able to migrate to the lymph node and process the viral antigens to be presented to CD4, CD8, and also help in the generation of antibodies. So they really are an important linker between this innate and adaptive immunity. So viruses that are able to harness these cells are particularly effective in establishing infection because they basically kill two birds with one stone. They're able to stop innate immunity so they can establish infection in the cell, but they might also be able to either delay or modify the generation of adaptive immunity. And this is just, we were intrigued of how dengue virus is really able to do that since it targets mainly dendritic cells for infection. And this is a picture of a human dendritic cell that is infected with dengue, and dengue NS3 is actually in red here. So as you can see, it's around the ER of the cell. So they do get infected and they have productive infection and release infectious particles. So I'm not going to go through this data because it's old data, but basically we were able to demonstrate that indeed dengue infects human dendritic cells, primary in the ex vivo. And then if these cells look activated, they're able to produce cytokines and chemokines. But one of the things we notice is that type 1 interferon is inhibited. So they secrete inflammatory cytokines and chemokines, but they don't really secrete type 1 interferon, including cells like PDCs. And the other thing that we found is that if they are infected with dengue, these cells do not respond to any other TLR ligand. They cannot get stimulated again by any other stimulation of the RLRs as well. And if we try to co-culture dendritic cells infected with dengue with naive T cells, they could not mount a Th1 immune response. So this basically tells us that infection by dengue renders these dendritic cells unable to induce a proper priming of the adaptive immune response. So maybe this is one of the problems in many infections, that maybe there is an aberrant or non-ideal adaptive immune response against the virus. And we found that the protein that was responsible for blocking type 1 interferon was the protease. So of course, this is very fortunate for us that we found that it was the protease, because if you have a viral protein that has an enzymatic function, it's much easier to really try to find out what is the mechanism, because it's likely that it's going to cleave some host factor or it's going to be affected some host factor. So we went ahead and started looking for what could be the host factor cleave. And this is the work from Sebastian Aguirre that is no longer in the lab, but he's a fantastic thinker and a fantastic researcher that really worked all the pathways and was able to figure out how this happened. So I'm just going to remind you that normally RNA viruses are sensed in the cells by RLRs, which are RIGI, and MD5, which are these cytoplasmic sensors for RNA, 5' triphosphate RNAs. And then there is this cascade of activation involving different factors that results in the activation of NF-kappa B, as well as IR3 and TDK1. And this actually results in the production of type 1 interferon as well as pro-inflammatory cytokines. And we also have DNA viruses that will be sensed through the sensors gas, and among others, but then it goes through STING to also induce the activation of IR3 and TDK1 as well as NF-kappa B, resulting in the production of type 1 interferons. So these are really the main sensors in the cell for viral infections, and actually the gas is also important for bacterial infections. But one of the things that we know is that STING also can cross-talk between the two pathways. So Sebastian went and tested the protease of dengue in overexpression systems together with all the components of the RIGI pathway, but we were surprised because we didn't find that this protease cleaved any of the components of the RIGI pathway. But interestingly to us, when he tested STING, actually he's using STING, wild type NS2B3, and we have a mutant version of the NS2B3, which is proteolytically inactive. So we see that when he tested human STING, we could see a cleavage product by the protease of dengue, but not when we use the mutant protease. But also the other thing that was interesting is when he tested the mouse version of STING, it was not cleaved by the viral protease. So we basically proposed that maybe not being able to cleave the mouse version of STING is actually one of the restrictions for dengue to infect immune competent mice. So we went ahead and we thought, as you know, was mentioned by Lina, why would an RNA virus care about a DNA pathway? But that would, why would we care about STING? And so we want to say, okay, we found it by overexpression, but we want to know what happens in real infection. So what we did is basically infect dendritic cells with dengue, inactivate dengue or mock, and we actually stimulated the DNA sensing pathway. So we wanted to know if these dendritic cells were able now to respond to DNA. And here is just showing you that this is mock infection, dengue, this is one of the viral proteins, and UV inactivated dengue is not expressing viral proteins. And we see that the moment that we see appearance of viral proteins, we see disappearance of STING in endogenous conditions, and then not in the UV inactivated virus. So then when we look at what was the result of this stimulus by E. coli DNA, if the cells were pre-infected with MOG or UV inactivated, they responded to this addition of E. coli DNA by producing interferon beta, interferon alpha, and also interferon inducible proteins. But if they were pre-infected with dengue, they were not able to really get stimulated any longer. So this really proved to us that the DNA pathway was also affected by this degradation or cleavage of STING by the dengue viral protein. So these are the conclusions. We found that it cleaved human STING and not mouse, and then endogenous STING was cleaved in dendritic cells, and this degradation actually blocked production of interferon in these cells. So we wanted to know, by then, when we were done with these studies, we actually is when CGAS was discovered. So we wanted to know, does the virus really even target CGAS, or is only just by targeting STING is sufficient? And as you know, CGAS would sense the DNA from viruses, protozoa, and bacteria, make these molecules called CGAMPs, which are the ones sensed by STING, and then the signaling cascade happens. So we went to monocyte-derived dendritic cells, and here I'm using 2 different serotypes of dengue, and I will talk about serotypes later. And then UV inactivated dengue, and you can see that CGAS is actually degraded pretty early during infection by both serotypes compared to UV inactivated or MOC, and then also STING. So they have different timing, by both CGAS and STING were degraded by dengue virus in infected conditions in primary cells. So we went back and we really wanted to know what was the mechanism of degradation of CGAS, and we thought it's probably going to be the same thing. It's going to be the protease that is going to cleave CGAS. And just to remind you, the protease is formed by the NS2B and the NS3. NS3 is also the helicase, which is important for transcription. It's very important for, it has many different functions, but NS2B is this cofactor that when it binds to NS3, it forms the protease complex. So we express the individual components with the complete NS2B3, NS3 alone, NS2B alone, together with CGAS. And we were quite surprised that NS2B, which is the cofactor, was the one where it was, when it was expressed with CGAS, this is CGAS flag, it actually degraded the CGAS. So we actually wanted to know what could be the mechanism. So what we did is actually here we're expressing NS2B together with CGAS, and then we actually blocked, so we see the disappearance of CGAS in these cells, but then we block different pathways of degradation of proteins in the cells, so cascade pathways like proteasomal degradation. But when we actually block the autophagy lysosomal degradation pathways, we see the reappearance of CGAS. So we recover expression of CGAS. So we believe that the degradation of CGAS is really not by cleaving through the protease, but by actually autophagy and lysosomal degradation of CGAS. So if you look at the autophagy pathway, we have different ways of blocking, and 3MA is used to actually block very early stages of autophagy. So we wanted to know how early can we block this and see the effect. And here is just in the context of infection. These are infected cells in the presence of CGAS. We see again the CGAS gets degraded. And when we add inhibitors of autophagy, we recover immediately this expression of CGAS. So it's very early during the autophagy. If we block the early autophagy, we actually recover the expression. So, and this is just quantification. So this really tells us that there is something, because viruses like dengue and other viruses really do have a very, very tight connection with the autophagy pathway. Many viruses induce autophagy early in infection to be able to steal components from the cells to actually make their own components. So maybe this is a collateral effect of the inducing autophagy that some of these host factors get degraded as well. And then we also were kind of intrigued because there were some seminal papers published. This is actually John Scoggins' work in Charlie Rice's lab at the time, in which they had done a big screening of different restriction factors for different viruses, and they actually found that cGAS, and here I put it with an arrow, this is the name of the gene, has shown a very strong restriction for many different viruses. Here I'm showing you yellow fever, West Nile, Venezuelan encephalitis, chikungunya virus. And then again, this is again showing you how strong this restriction is. But when they actually plot these restriction factors that they were testing, that were some of them ISGs, they found that cGAS has the strongest restriction for positive strand RNA viruses, which are in blue, but no restriction for the negative strand RNA viruses. So again, there is something about these positive strand RNA viruses, the way they interact with the cell, that maybe is result in this induction of cGAS or activation of cGAS. So we were intrigued by this, and we wanted to know, first of all, where other viruses could be, if in other viral infections we see this degradation of cGAS, and also what it would be the mechanism. So we went to chikungunya virus, which is not a plague virus, it's an alpha virus, but is also transmitted by mosquito, by the same mosquito. And in chikungunya virus, I don't want to really get too much into details, but we didn't really look at the dendritic cells because the dendritic cells play a role at the beginning, but most of the disease is caused in the joints. It causes chronic arthritis. So we wanted to find some epithelial cell that we could use that was also primary. And I just want to show you that most of the cell lines that you will encounter to do overexpression experiments really do not express cGAS or STING. So they do not express endogenous cGAS or STING, which is fantastic for overexpression experiments because you can express anything you want. But you really, if you want to do endogenous, look at the endogenous levels, you don't find it. So here we found these human foreskin fibroblasts that are primary cells that do express both cGAS and STING, and they're infected by chikungunya. And we actually did the experiment, similar to the experiment that we did with dengue. This is mock infected HHF1s, chikungunya infected. We see a chikungunya viral proteins, and we see that it also degrades cGAS very early during infection. So cGAS is also degraded in chikungunya infection, but to our surprise, STING was not cleared. So again, I'm not going to go into details, but we find that viruses might interact with proteins and not cleave them, but actually sequester them. So this is one of the pathways we're looking into now. But we see that cGAS is also degraded by chikungunya. And again, just to show you the same methodology we used before, we block different pathways. We express cGAS in chikungunya infected cells. And then we look at endogenous cGAS, and then we actually found that if we block autophagy, we recover expression again, but not if we in the vector. So we actually see that, again, it's through an autophagy-mediated mechanism that cGAS is degraded in chikungunya infected cells. So we know that the RNA viruses really have many pathways and many ways of blocking the read pathway. In dengue infection, we could see that it cleaves cGAS or eliminates cGAS, STING, and also cGAS. So we wanted to know what was triggering cGAS. And so what we did is actually going back to our 293 T cells, which are really not my favorite cells, but they're really good for doing overexpression. The reagents for cGAS are not that good, so we could not really plan this experiment with endogenous cGAS at the time. But we infected these 293s that express cGAS with mock or dengue. They pull down with an anti-flag, so we pulled down cGAS and we did DNA extraction and characterized what was the identity of the DNA that was bound to cGAS in this infected cell. And we sent it to be bioanalyzed, and then we actually found different bands. But this one was particularly strong, and we thought, it would be really worth sending for analysis of this sequence. But in the meantime, Sebastian was really very eager to know what could be the DNA, and he actually designed different primers, and he looked at different genes or different DNAs that he could define that are actually from cellular genes. And then there was really no real pattern between mob or dengue infected cells in terms of the DNA that could be bound in cigars from those genes. But when he looked at mitochondrial genes, this was actually very clear that there was an enrichment of mitochondrial DNA bound to cigars in these infected cells and not in the mob. And when we got back the results from our deep sequencing, we actually could confirm and it validated our data that there was a big enrichment of mitochondrial DNA bound to cigars in the infected cells. So we also, this is work by Tonton, that is a student in the lab, and we actually had infected cells, and these are the viral proteins. This is then the fluorine-4. And then we actually could see this DNA that is actually leaking into the cytoplasm. So we actually can detect leak DNA, which is different from DAPI. And then when you do the merge, we actually find that they are all in close proximity. So they are cGAS, dengue proteins, and the DNA are all in close proximity in infected cells. So then we found that cGAS gets degraded in dengue infected and also in chikungunya infected cells. And the dengue inhibits the production of interferon in human disease by blocking this pathway, cGAS sting, and then is mediated by the NS2B through this autophagy lysosomal dependent mechanism. And we found this enrichment of mitochondrial DNA that we believe is triggering the pathway. So it's basically the cells are really sending this danger signal, these dumps that are not necessarily the genomic material of the virus, but it's really a response to the infection that actually is triggering the interferon pathway. So now the viruses have to come up with strategies to block this pathway. And then one of the things that we wanted to see is like how does dengue induce this leakage of mitochondrial DNA? We went back to the literature and we actually found that there had been some of the viral proteins already being described as having some role or some interaction with the mitochondria. So for example, PM has been shown to create pores in the mitochondria. Also the NS4A localized in the mitochondrial membrane. And then that affects MAVs in signaling cascade, and also NS2B3, the protease can localize in the mitochondrial membrane and cleave mitofusin. So these already have been shown, but we had not made the connection of the two consequences of this. So I now want to go change a little bit the direction, because we did all these things in vitro and we were very excited about finding this mechanism, but we actually wanted to now basically validate or look at what happens in dengue infections in vivo and what do we see that could be interesting to study. So here I show you the flavivirus family, and it has many viruses that you're probably familiar with. They're all mosquito-borne here. And in the mosquito-borne, you have West Nile, Kunjin, San Luis encephalitis, Japanese encephalitis, the 4 dengue serotypes, yellow fever, among others. But the dengue viruses have been divided in 4 different viruses that are called dengue 1, 2, 3, and 4, but they're really very different viruses. So they could have been called different names, but they're really all called dengue, but they cause different severity of disease. And we wanted to validate if what we have seen was actually true for all the serotypes, and also if we use primary isolate, we see the same thing. So this is something that we knew from the field, that dengue 2 is mostly associated with this more severe disease, and in particular in secondary infections. And dengue 4 is really very rarely associated with severe disease. It causes mild clinical presentation. And this is work by Rebecca Hamlin in my lab, that was an MD-PhD student, that she actually infected dendritic cells, and this is virus released from these dendritic cells to the supernatant. She could see that dengue 4 in purple actually peaks, the release of particles peaks very early at 24 hours, but then it starts declining. But dengue 2 actually goes down and then starts creeping up and peaks. So basically the virus released by the 2 serotypes are very similar, the amounts over the course of infection, but they peak at different times and they have very different kinetics. But then Kevin Maringer in the lab actually looked at the same infection in mosquito cells that are actually not deficient of any of the immune pathways, and there was really no difference in the kinetics. So there is something about the human system that made these viruses replicate differently and produce virus peak at different times. And then we look at the cytokine release also in the supernatant, and this is again dengue 2, dengue 4, and MOG, and we can see that actually every cytokine that we measure, TNF, IL-1 beta, including type 1 interferon that we don't see in dengue 2, we actually could see that dengue 4 induced this secretion of these pro-inflammatory cytokines, including type 1 interferon. So we were really intrigued by this, and we wanted to know what could be the mechanism. And Gabe and others even in the lab, really what he did is clone the different proteases of the different serotypes. And here we're testing, this is a mutant protease, this is the dengue 2, dengue 3, and dengue 4 proteases. And then we also did the yellow fever, Zika, French Polynesia, and we actually expressed with human sting these different proteases, and we could see that all three serotypes that we tested here actually cleave sting, and dengue 4 that induces production of interferon actually cleaves sting even better than the other two. So that was, and also yellow fever didn't, or Zika didn't cleave sting, but they actually, yellow fever interacts with sting. So we really could see that this is not the reason, or doesn't explain why dengue 4 induces production of interferon. So we actually, at that point, we were already part of a big project that I will talk about in a minute, that allow us to really look at these much more sophisticated immunology techniques that I am in love with, because I was really able to now extract much more information for our samples and from our samples. And we started using cytos that probably for this audience, I don't need to explain, but it's really allowing us to use 45 parameters in the same sample without the need of compensation, using a big panel that we can apply to many different samples. And we only need 10 to the 6 cells or something like that. And by using the heavy metals, it really doesn't reach into the channel. So it really is much less difficult to use, at least to have this bleaching between the channels and to really define the different expression of the markers. So we went to our dengue infected cells, and this is actually in dendritic cells. I think we use 18 or 20 markers in this case. And this is two different antibodies that recognize viral proteins. The 42 recognizes the envelope. E1D8 recognizes the NS3, which is a non-structural protein. So we were looking for cells that express both viral antigens. So this is in dendritic cells, as I mentioned. And one thing that, and this is dengue 2 infection versus dengue 4. Using this technology, we actually found two different things. One of them, dengue 2 infects very few cells compared to dengue 4. And also they are more bright. So the cells that are infected with dengue 2, although they are less cells, they really express probably more antigen. But then dengue 4 is really infects a massive amount of cells in the culture. But the thing that was also interesting to us is that what happens to these bystander cells in the same culture? Can we really look at what other things are they expressing? Can we really, using cytos, now we can really gauge in all these different markers. We were actually staining for cytokines, chemokines, and activation markers. So we really wanted to know what happened to these bystander cells. And here I'm showing you TNF that we measure intracellularly. And this is again dengue 2 in green, dengue 4 in purple. And if you look at MOG and bystander dengue 2 and infected dengue 2, in every case, and also here is dengue 4, the TNF is really released by the infected cells, not by the bystander cells. So we see that they have different kinetics, but the infected cells are the ones secreting or producing the cytokines. But we went now to another cytokine, which is IP10, which is actually induced by interferon, induced in the same way as interferon, but also is an interferon-inducible protein. And we can see that actually the bystander cells are the ones producing IP10. So it's telling us infected cells can control the interferon production very well, but then if there is a leakage of some of the signaling or some of the molecules, the bystander cells will not be able to block because they don't have viral proteins inside, and they are now producing interferon or interferon-inducible cytokines. So this is quite important for us because this virus doesn't really infect that many cells. There are very few cells infected, but we can now see the contribution of bystander cells and infected cells to the infection. So we actually now see that the different serotypes induce having different infection kinetics in these dendritic cells, and also all the serotypes we tested could degrade sting. But by using cyto, we can really see now this contribution of the different cells in cultures. at a single cell resolution, and we can really look at the bystander cells. And this is actually a very valuable tool for not only for our ex vivo studies, but also for looking at clinical samples in which you don't have that much availability of cells that you can run in flow, for example. So we wanted to now kind of improve our model a little bit more, and then we went to this human tonsil because we have access to tonsils that are removed because of mostly sleep apnea in healthy donors, and we actually is basically representing the lymph node stage of dengue infection, and it will be a novel model for dengue. So this is work again by Rebecca, and she actually was this is the way the tonsils look. She can cut them into pieces, and also now I have other people in the lab doing this. We can put them in collagen beds that have 9 blocks per collagen sponge, and we rest them overnight, and we can basically add the virus on top. So it's like a lipid interface, and we can now measure different things. We can measure by collagenase treating the tissue. We can do flow cytometry, cyto, RT-PCR for like different markers. But we also can do frozen or paraffin sections with photon microscopy. But in the culture, in the media, we can actually look at virus replication or how virus release, but as well as cytokine by ELISA or multiplex ELISA. So this is just showing you 20 different donors in which we had infected tonsils, and this is different actually input of virus, and it's very difficult to calculate how many cells are going to get infected. But regardless of the input, we could see that from day 3 to day 6 post-infection, we can see a nice increase of virus replication that it actually keeps increasing or maybe plateaus, but we see a very, and this is actually virus release to the media. So this is replication intracellularly that we can now detect in the media surrounding the tonsils. So there is a productive infection in the tonsils. And then we went and we measured this is actually days, not hours. So by day 2 to day 6, we really cannot detect any type 1 interferon, which actually validates what we have seen in dendritic cells, that there is very little production of interferon. And probably for dengue 4, this is too late to actually detect type 1 interferon. But then when we go to IP10 again, we really see production mostly by dengue 4. But also by day 6, it's already in all of the samples we see a little bit of IP10. But the thing that is also consistent is that we see now the production of more Th1 cytokines with dengue 4 and not dengue 2, at least higher with dengue 4, which might implicate that by day 6, day 4, we see the release of cytokines by other cells, such as T cells, that are now responding to this infection in a different way than the ones infected with dengue 2. And this is now showing the TNF as well. So we can use these tonsils. We can now measure different things in the tissue. We are doing multi-parameter flow cytometry as well as cyto. And we really now are trying to understand these interactions between the different cells, the infected cells. We're trying to find what the infected cells are in the tissue. So this is being quite an exciting system for us. We observe different cytokines and chemokines, so we can really measure, and it's reproducible despite the donor-to-donor variability. So we see that dengue is productively infecting these tonsils, and this is now a model that we are trying to establish for dengue. So at that time, I was able to really put together a big project with Eva Harris that has a fantastic cohort of dengue infection in Nicaragua. And we wanted to really ask the question of what are the factors that determine the severity or the outcome of disease with dengue. And in particular, I was very, very focused on the innate immune factor. So we wanted to capture early events and early interactions that might determine the outcome. So we have 3 projects: natural infections of dengue. We wanted to study them from the cohort that Eva has in Nicaragua. Also, we have access to the vaccine trials of Anna Dubin and Steve Whitehead with the tetravalent dengue vaccine. So we wanted to really look at the response in the vaccinees. And then in my lab, we're really doing these ex vivo infections of PBMCs, dendritic cells, but with the viruses that we obtained from Eva's cohort as well as the vaccine viruses. So we also have a fantastic set of cores that assist us with with this, Steve Wolinski runs the genomics course. So we're looking at transcriptomics. We are sequencing all the samples, looking at maybe genetic factors that might determine the outcome, as well as proteomics with Nevan Krogan and Jeff Johnson, and then the immune phenotyping that Mike Birian and Adib Rahman in her group that has really allowed us to now also combine the data between the natural infections, vaccinations, and ex vivo infections. But then, of course, nothing could be done if we don't have good data analysis cores that are really now to put together this data. And I just don't want to spend a lot of time, but I want to show you a couple of examples of what we're doing with this project, which is, for example, here I'm showing you dengue 2 infection in PBMCs compared to dengue 3 or dengue 4. And if you look at the cytokines, this is Luminex data, in which we can see that just by looking at it, they're much more, the red is higher expression, there is much more expression of inflammatory cytokines in dengue 3, dengue 4 infected PBMCs compared to dengue 2. So in PBMCs, we see the same features we see in the dendritic cells and in the other systems, that dengue 2 resembles more the MOC, with the exception of interferon-inducible proteins like IP10 that at late times, this is 48 hours here, we start seeing that appearance. And then we can also do cytoph in these PBMCs, and this work done by Rafa in the lab, in which I don't think I have to explain to this audience either, but we can really like look at this expression of all these different markers that actually define the populations, and we can actually find maybe some populations that are not defined by the classical markers, but using cytoph, and this is actually 40 markers. We were getting in the myeloid cells, we know are the target cells for infection. And this is CD40, for example, expression of CD40 in the MOC. One thing that was important, we have to have a MOC for each time point because the cells change over time in culture. So we see a little bit of activation in culture, but actually when we look at dengue 2 infected cells, that's where we see really strong activation of the myeloid cells. So we really can see that although dengue 2 seems like very silent, it's also inducing changes in this myeloid compartment. So we now can go and get, on the different cell populations and look at the difference between the serotypes. And this is, for example, RNA-seq data from dengue 2, dengue 3, dengue 4. And the main difference here that at early time points at 6 hours, we see like these classes, these actually are pathways of innate immunity, like viral sensing, innate activation, different antiviral interferon signature. And they are very, very like low expression in dengue 2 infection at very early time points, that it actually goes up over time. But actually in dengue 3 and dengue 4, we see a much higher expression of this gene. So it's really activating these things very early on. And these actually, importantly, we have to do these ex vivo infections because we cannot capture these in the cohort. We really don't have 6 hours post-infection. So the goal is to really combine what we can do in the ex vivo infections compared to what actually the outcome is in this natural infection for vaccination. And in the vaccinations, we have a little bit in between because we have day 1 of vaccination in which we can analyze the sample. So we actually see that dengue infects and induces phenotypic changes in myeloid cells in PBMCs. We have lower signature of dengue 2 compared to dengue 3 and 4. And then the Nicaragua, this is actually viruses from Nicaragua, which are primary isolates. And in dengue 4, we see are the only ones that induce type 1 interferon. And early in infection, we see this lower immune gene expression in dengue 2 again. So this is really kind of coming back and wrapping up. It's telling us that we really need to look at how each serotype or each virus is really having a combination of strategies. So we believe that dengue 4 is, we know it's very early in terms of peaking, the replication is fast, so it causes probably early viremia. So that might actually result in very early transmission by mosquito. And this is at the expense of inducing a strong innate immune response in humans. And then this also results in probably a very strong adaptive immunity of the Th1 type that results in early clearance of the virus and maybe less severe disease, like it's been seen in the field. And I actually call it like maybe it's like being vaccinated. So you have very mild disease. And this is what normally viruses like to do. Viruses don't want to kill the host, but dengue 2 might be less adaptive. So it actually requires longer to peak. So it causes late viremia. So it needs to tweak more elements of the innate immune response in order to do that. So that results in late transmission to mosquito. but then it actually has a very low profile of innate immunity, but also a very low, maybe or different production of the elements of adaptive immunity that results in the late clearance of the virus that might actually result in more severe disease. So this is our model, how to explain, but also to kind of emphasize that there is not going to be one single strategy by a virus, but it has to have an equilibrium of maybe inducing a stronger response, but it has to have more tricks to block it. But at the end result is that the viruses want to make more copies and transmit to the next host. And then I just want to end with my lab, of course. And this is a fantastic group of people. And this is a combination of virologists and immunologists, which I love because it's really learning from each other and being exposed to the different approaches, techniques, and ways to work that is really making it fun. And now the immunologists are not afraid of Western blots and the virologists are not afraid of flow. And that's kind of the goal of my lab. And I really am very fortunate to work with this fantastic group of people. But also have great former members of the lab as well as collaborators, and also like Adolfo and his team, but also the HIPSI grant. The team of HIPSI has been instrumental to really do a lot of the validations in vivo, and of course the funding of my lab as well as the funding to my students has been great for doing all this work. And I just put again the names of the people in HIPSI, but it has been a fantastic ride with them. Thank you.
Thank you so much, Anna. That was a terrific talk. Yes, thank you. Well, I just type a question in the chat, and I don't know, Matt, if you have a question, you can go first.
No, why don't you go, Lena, and I'll hold my question until the end.
Okay. So we'll give more time also for people to ask questions. Yeah, so Anna, this was great, fantastic. So if I understood correctly, dengue 4, you show that it peaks earlier in terms of viral production of virions, right? It also infects more cells in the dendritic cell cultures, and also induces higher cytokines, interferon, and I think TNF you showed. But what is the causal relationship between all these phenotypes? So is it that it produces more interferons because it infects more cells? If you normalize to the number of infected cells, is still higher the interferon production?
We are looking into that, but one of the things that is very characteristic of dengue 4 is that the polymerase is very fast. So it really replicates very fast. So I think it really creates too much pump. So it's a virus that is replicating so much that it cannot catch up with actually now blocking because it's producing too much of the pump. And I think it reaches a point in which it can really, there is more pump than actually what the virus can do with and then some of these proteins are also needed for replication. So I think is really like pushing the system a little bit. So the goal is probably replicating very fast, but it's at the expense of maybe inducing more innate immunity. But by the time this interferon response is happening, the virus is already gone. So it doesn't really care because it's not going to affect the overall production of virus particles. So it's maybe late for the virus, but then that results in a better adaptive immunity. So then next time you encounter dengue 4, you're more protected. So dengue 2, so it's almost like a vaccination. It's really like using the kind of the best part of the innate immune system, but because it's so quick, it just escapes before the actual consequences of the innate immunity kick in.
Really? So you think that the driving force is the increased activity?
Yeah, and I think we're now looking into subgenomic RNAs and other things that are very different between dengue 2 and dengue 4, but also something that we want to look into is like what, the dengue 2 cells, infected cells were lower, but they were very bright. So they also probably produce more viruses per cell. So one of the things is that we don't know what's going on in the infected cells with dengue 4 versus dengue 2, that maybe, is more efficient in dengue 2, but it takes longer. So there's this kind of game of the equilibrium between being a more silent virus, but you need to really have more tricks in your bag, But if you're a very like a bull in a China store, but you don't care what you break, but you just get out. So it's really a little bit of like that dynamic and the kinetic. So taking into account everything, the speed, the efficiency of replication, as well as how much of these pumps are generated during the infection of the tube.
Yeah, cool, perfect. Thank you, Anna.
Okay, so since no one else has any questions, I will ask my question super fast. I realize we're running out of time. So Anna, going back to the first part of the talk, you showed both of these viruses, both chikungunya as well as the flaviviruses, actually result in autophagy-mediated C-gas degradation. So what's known about C-gas sort of steady state homeostasis in terms of, is autophagy playing a role in the natural turnover of C-gas, and are the viruses basically just activating this normal homeostasis mechanism, or is there something specific that the viruses are doing to sort of plug C-gas into the autophagy pathway? Do you know?
Yeah, this is the T32, not the T32, the F31 of my students.
Okay.
But we're looking into that. And I think we know that the virus induces autophagy, and we believe it's NS2B, which is the same protein that interacts with C-gas.
Right.
So I think the protein is bringing C-gas to the autophagy machinery.
In a specific way.
Yeah, but we don't know if C-gas by itself, like you say, it also needs autophagy for something. But I think because everything is such close proximity, because all these proteins are in the ER membrane, right, and the mitochondria and the ER membrane, and it's just all so close together, and these have coordinated immunity. So I think, and if you see, for example, flavivirus and alphaviruses, they make these viral factories, so they need a lot of membrane. So they really need the autophagy mechanism to create these kind of vesicles for replication, because one thing that is also cool about these viruses is that they make these vesicles for replication, so they hide the RNA from the RNA sensors. So I think that's why they are so efficient at hiding from the RNA sensors, that now the cell is sensing something funny and is releasing this other danger signal that now is inducing the pathway. So it's this constant battle between, you master one pathway, and now the cell is throwing a wrench at you, and by inducing it in a different way. So, and then the virus has to come up with a way of blocking that. So I think it's really exciting and fascinating to see this kind of dynamic of how they have to constantly adapt to each other. But yes, I don't know if C-gas goes to autophagy normally, but that's something that has to be looked at. Absolutely.
Cool. Thank you. So we are past 10:30, so I guess we will move on and thank Anna for a really nice seminar. And she will now go off to individual meetings. So
thank you. Sorry, maybe I went over time. I hope not.
No, you were perfect.
Okay. Thank you so much, Anna.
Thank you.
And so, now we'll proceed with Angelo from BioLegend that will be sharing with us some of the newest reagents. New ones are not so new. I don't know, Angelo.
So mostly new.
Mostly new reagents that BioLegend is offering for research technology. So let's do that. And thank you so much, Angelo, for the efforts to prepare that for us. Thank you.
Absolutely. All right. So thank you for the introduction. I think, can everybody see my screen, I think? All right. Let's rock and roll.
Yes, Anna, you may project it. Yeah, exactly. Perfect.
Okay, great. Thank you, everybody. All right. So let me move this around a little bit so I can see my screen. Okay, great. Well, thank you for the introduction. I'm very happy to tell everybody about some of the newer things we've really been doing here at BioLegend recently. Of course, a lot of the digital outreach we've been doing and connecting has been great. I miss meeting everybody, so hopefully we can do that kind of soon. But today I'll be telling you guys about emerging technologies and platforms for legendary discovery, kind of just glazing over some of the more newer things that we have at BioLegend. First, our central mission is to enable legendary discovery from research to cure. And that was a vision that our founder and CEO, Gene Lay, had. And it rings true with everything we really do here at BioLegend since our incorporation in 2002. We have our headquarters here in San Diego. We have a beautiful new campus. We actually won some recent awards for our campus and our buildings, and hopefully I can extend an invite for tours in earnest sometime soon, but we'll have to hold off on that for now. Collaborations and integrations with several academic institutes are a huge part of what we do at BioLegend. It can't be understated. So I really do appreciate everybody listening here today and looking forward to continuing collaborations with UCSD in particular. Because I went there, it's my alma mater. No, every institute that's listening here, I'm very happy to be here and be reconnected with some of the researchers in the area. Okay, so one of the ways to see how we are doing as an organization and towards our mission statement is to look at our impact on the publication field. And as you can see here, BioLegend is the most cited antibody supplier for immunologists performing flow cytometry, which to me is a nice indicator of how well our technologies and platforms are doing out in the wild. Okay, so we are historically known for being an immunology company that for flow cytometry antibodies, but we really have since expanded our catalog to include many different disciplines. And today I'll just be glazing over 3 new things that we do have and new technologies to help with some of the research today. So first, I'll be talking about spectral flow cytometry. It's sort of a new technique compared to conventional cytometry. And so I'm going to briefly touch on the differences in conventional and spectral cytometry for the audience here today. We have PE, the PE fluorochrome here. It has a blue laser and a yellow-green laser excitation, as shown here. And in conventional cytometry, you have to read this light signal somehow. So what we have here is we have a channel as an example here, and you can measure the PE signal here. Now, one of the major differences is that in conventional cytometry, instead of looking at just a little piece of the emission wavelength here, you're looking at the entire fluorochrome, the entire signature for each laser. So what you get are graphs that are down here below. You look at all the emission from the blue laser, the yellow-green laser, and the red laser. This is a 3-laser spectrum instrument configuration here, for example. And so what you get is after you knit these different signatures together, you get a full spectrum profile or fingerprint, a unique spectral signature. So here in PE, because we have excitement on 2 lasers, you have emission on 2 different lasers, and that's seen very nicely here. So in practice, what that allows you to do, because you're using unique fingerprints of each fluorophore you're using, it allows you to combine them with a little more ease. And also compensation isn't as challenging with in comparison to conventional cytometry. So we have 3 fluorophores here, or rather 3 major classes and families of fluorophores: the simple organic fluorophores, fluorescent proteins, APC and PE are examples. And then our 3rd class here, which are polymer-based dyes, and they are relatively new. Brilliant violets are an example of that. And today I'll be telling you guys about a new fluorophore we've been developing in collaboration with Sony. And these are our Caravia dyes. So we've also developed spark dyes, which are simple organic dyes, and our fluorescent protein tandem dyes, which are our fire dyes. So let's start off with the Caravia dyes because it is a new novel dye chemistry. First of all, Caravia, it's an interesting sounding name. It's actually Japanese for sparkling or shimmering. And it's a well-named molecule. If we look at it here on the right, here's a quick little example of this new type of chemistry. So what you can do is you can load onto this backbone all these different fluorochromes and different other chemistries, and that allows you to fine-tune and customize the characteristics that you want. So really excited for this new fluorophore family. And today I'm going to be telling you guys about Caravia Blue 520. So right off the bat, we have to talk about Caravia Blue 520 and FITC. And so the reason why I chose this dye in particular, because it can be used in conventional cytometry and spectral cytometry. So it has the exact same spectral signature as FITC, and I'm using a 5-laser orientation example here just to kind of give you a big holistic approach here and looking at the spectral signature. So some people say, well, you know, it's the same thing as FITC. What advantages does that have? Well, in conventional cytometry, let's just say you have a very limited number of channels you can use in your instrument, or even if you really don't want to move your panels around too much. You can put Caravia Blue on other markers that would maybe have more disparate distribution or intensity, where normally you couldn't put FITC on. So for example here, if you look at some of the stability data in comparison to FITC, Caravia Blue is significantly more stable here with our light dark test and exposure. And also here is an example of putting Caravia Blue 520 on an intranuclear stain, rather an intranuclear marker being Tibet. So it's a really nice alternative in the conventional cytometry space where you can put Caravia Blue 520 on markers you normally would not be able to do, say, in the FITC channel. The second new fluorochrome we'll be talking about today is our APC Fire 810. This is a really popular fluorophore since it's released. It's available now because it's a new channel. It's a brand new fluorophore, so it can be added to most existing panels. So let's look at some of the graphs here. Again, I have the 5-laser orientation for the spectral signature, and I'll be using APC Fire 810, which is a far right infrared emission here, APC Fire 750, and Zombie near infrared. So let's look here at the graph on the left, though. This is the conventional spectrograph that you would see. And as you can imagine, you look at that and you say compensation is going to be challenging and really might be hard to use these fluorophores in tandem. But because, again, we're using the full spectral signature of each fluorophore, these little arms allow you to be able to use all these different fluorochromes in tandem, which would normally be challenging in conventional cytometry. So we also, to demonstrate some of the staining data, we put APC Fire 810. Though it is a tandem dye and is a bit larger, we still get really nice staining with intranuclear markers as well as, intranuclear markers in Tibet, as well as intracellular markers in interferon gamma on CD positive cells. Okay, so here's an example of all the different multicolor capabilities you have in a 4-laser instrument. In purple, we have all the fluorophores that are available at BioLegend now. The red ones here are new fluorophores we are releasing. Unfortunately, for the sake of time, I was only to really highlight 2 of them, but we have more fluorophores in development to try and fill in these little spaces here to really get the plexity of spectral cytometry. So next I'm going to be using, discussing Legendplex, which is our multiplex analysis of soluble targets by flow cytometry. So I think the discussion we had before this with some of the chemokines and cytokines was a great sort of segue into this technology. So Legendplex is more commonly used to measure cytokines and chemokines, which are really impactful for biology. To demonstrate that, here we have an example where naive CD4 cells can be exposed to different recombinant growth factors, cytokines and proteins to control the differentiation to other different cell types. And we also do carry all these recombinants at BioLegend. Cytokines have also been in the news and kind of in vogue recently because of its implication in coronavirus. So I was actually watching TV, or had it maybe on the background one day, and I think we're all used to seeing Anthony Fauci on our TV screens and our computer screens, but he was giving a talk to, I believe it was the Senate of the Congress, and he was referencing a paper, a Cell paper. I was able to recognize this paper. This was a really nice article and project done by the Karate and SETI labs looking at T-cell responses to SARS-CoV-2 infected patients. And so they actually used Legendplex to analyze some of their patient samples here. So it was a nice kind of, props to LJI for doing this work and a nice way to show the utility of Legendplex in the context of SARS coronavirus. Normally, if you just use an ELISA, for example, I think it's a very common approach we're used to. You use one sample, you look at one target at a time. However, if you're looking at multiple targets at one time with just one sample, it takes less time, fewer reagents used, and therefore it's more cost effective. But really, one of my favorite things to point out here is that it's lower sample required. You only need this one sample volume to measure all these different targets at once. And so if you have very precious samples, patient samples, clinical isolates, this is a really nice option to look into. Briefly, I'm going to talk about the assay principle here. Instead of having an antibody coded to a plate, like in an ELISA, for example, we have the first capture antibody conjugated to a APC encoded microsphere. We then come in with a second antibody that recognizes a second epitope. This antibody is biotinylated, and then we can harness the profound affinity between streptavidin and biotin, which is conjugated to PE. And so we can use the PE signal to also as a surrogate measure for the target of interest. So let's look at what this looks like from a data acquisition side. For me, I'm kind of a more visual person, so looking at the data like this kind of helps out understanding what's going on. So in the Legendplex, we have our first 2 parameters here are different bead sizes. So we can see this here in forward Saturn side scatter. We have bead A and then bead B. And so they're 2 different sizes. So hopefully you can appreciate their disparate clusters here. So here we have an example of what happens when you have a Legendplex system running with the increasing analytes. On the y-axis, we have the APC channel intensity, and then the x-axis, we have PE. And hopefully you can see here, every little cluster, every little horizontal line is one bead, which recognizes one target. So as you increase the analyte concentration, as you'd expect, you see a shift to the right. And so this is how we can use a flow cytometer to measure multiple targets at once. So we have 300 plus targets you can look at, and our kits include anywhere from 3 to 13 targets per assay. So here's a breakdown of the different species specificity, most of which are human. We also have mouse, rat, and primate. And again, in some cases, you can get by with 12.5 microliters per sample, and even less if you have to dilute your sample. So it's a really nice way to get, answer as many questions as you can with a small sample type with a, the workhorse instrument immunology, which is a flow cytometer. And we have a few different flavors of LegendPlex available. We have our predefined panels, which are biologically focused, and it's anywhere from 3 to 13 different targets. But let's just say you don't want all 13 targets in your assay. You can then use our mix and match option where you pick cytokine number 2, cytokine number 4, and so forth and so on. And what's great about these is these ship next day if we have it, these two options here. And then if you need a little bit more customization, we can of course look into our custom solutions. And quick little plug, we have custom solutions for the vast majority of different catalog items and platforms we do offer. So if you encounter a reagent that doesn't exist, please reach out to me and we can see if we can do some custom assays or custom solutions for you. Okay, so I will spare the validation for you with the LegendPlex, but I will say that it is validated for intra as well as inter-assay precision. And what's great about that is all the data is included in the validation manual. So very often if you're submitting a grant or perhaps you're presenting a new technology for a manuscript, some of the validation data is required and it's right there. It's available for you to use and look at and use in reference. Very quickly, let's just look at how the LegendPlex performs by comparison to ELISA. This is a quick little heat map that I did, comparing ELISA, PBMC is exposed to these different stimuli in ELISA. And then here in the bottom, we have LegendPlex. And you can see that the profiles are fairly similar, but in some cases, for example, IL-21 here, you can actually get detection, whereas in ELISA, you can get no signal. So it has an improved dynamic range, anywhere from 1 to 10,000 picograms per ml per analyte. And in some cases, it can be more sensitive with certain analytes by comparison to ELISA and even the Luminex technologies. So quick little takeaways here from LegendPlex. It's multiplex detection on a flow cytometer, use less sample. One thing I'm going to point out here, we have a free cloud-based software, which is really great. It's free and it's also really user-friendly. And one of the great advantages with that is, you're analyzing your samples. Let's just say you have maybe a question of the analysis. You want to say, how's this looking? You can grant myself or my team access, and in real time, we can go in, look at your data, of course, when you grant us access, and look at your data and analyze it and sort of give your opinion. So it's a really nice added tool to try and meet, the needs of the end user. And also these are biologically focused panels, and everything's in the box here. Everything's included in the kit, as you see here in the bottom photo. Okay, so lastly, going to talk about TotalSeq. No emerging talk is really complete without talking about TotalSeq. This has been a huge area of investigation, and we actually have a few people possibly on this call that have been helping us out with some of our optimization. So TotalSeq, it's a little bit of a complex concept here. So I'm just going to take a quick step back and discuss, the overall use of TotalSeq. So first of all, as we all know, looking at RNA or how a cell makes its RNA gives a great insight or inference into the biological processes. And that's why RNA sequencing technologies have been so popular recently. And so we first had really RNA bulk processing, which is demonstrated here on the left. Great technique to look at what's going on in cell populations. But let's just say, for example, we are interested in a specific transcript here shown in red. With bulk RNA processing, you really cannot tell if that transcript is coming from one cell or all the other cells. However, with single cell RNA sequencing shown here on the right, we can normalize and we can say that each individual transcript comes from a single cell. And so this allows for very deep interrogation of cellular mRNAs, tens of thousands of genes and thousands of cells, as you can look for a lot of creative approaches to understand biology and really interrogate what's going on in your sample types and your hypotheses. So even though looking at mRNA, the transcript, what is a cell making into an RNA, is a really powerful technique, there are still some challenges. To demonstrate that, here we have a t-SNE plot. And so a really quick way of looking at this, the closer the dot, the more transcriptomally similar the cells are. So each dot is a different cell. Now, unfortunately, what you still have here is you have a clustering based on RNA of cells that look the same, but really we know have very disparate phenotypes and functionality. For example, we have CD8 and CD4 T cells clumping together and also different fibroblast lineages. So even though we have RNA sequencing, it is still difficult to distinguish transcriptomally similar cells. Again, we're just looking at RNA. But what if there's a way to look at RNA and protein at the same time? In other words, two different arms of the central dogma. And we can do this now with a technology called CytSeq and TotalSeq, and I'll make the distinction with those two things here in a minute. But let's just talk about TotalSeq, which is the BioLegend platform. So instead of having an antibody, which has its profound affinity and specificity for its target, instead of having it conjugated to a fluorophore, which we're commonly used to in flow cytometry, it's conjugated to an oligonucleotide instead. And so each antibody has its own antibody barcode. And so that's the unique marker for the protein, if you will. And then to mimic mRNA transcripts, we have a poly A tail here so that this can be incorporated in all the normal single cell RNA sequencing pipelines. So again, we have an oligonucleotide tagged antibody for protein detection. And so what this allows you to do is you add these TotalSeq antibodies and they bind to the surface of the cell, different targets, of course. And so while you're doing your transcriptomic analysis, you can then incorporate this, which is a really powerful technique. You're getting a really nice granular insight into what's going on in your cells. So to demonstrate that, I'll take a step back and again, make a clear distinction here. CytSeq is the name of the method and TotalSeq is the name of the BioLegend platform and technology. So here on the left is another t-SNE plot demonstrating if you cluster just on RNA, we have difficulty distinguishing CD8 and CD4 T cells here as an example. But if you cluster on RNA and the oligonucleotide from the protein marker, we have much better separation between CD4 and CD8 T cells, monocytes and dendritic cells. And also these islands are much less diffuse. They are much more centralized. So this gives you a little bit more confidence in some of the data that you're making. Again, really laser beam focus on what's going on in your sample types in an unprecedented way. So we have many different options for TotalSeq. I'll briefly just talk about, we have both 5' as well as 3' sequencing technologies for all the different single cell RNA sequencing platforms, but also we have some options specifically for 10x pipelines. So quick little review here. TotalSeq allows for simultaneous proteomics and transcriptomics from a single sample. And so this allows you to look at differential patterns between RNA and proteins in a way we would never really have been able to before. And so this allows for deep interrogation of sample heterogeneity. And so PBMCs are a great sample type to use to look at this because, as we know, they're very heterogeneous. The markers are distributed in many different ways, depending on the day, perhaps even. So we use PBMCs for all of our optimizations for TotalSeq right now, but we were constantly looking at new sample types and sources. But PBMCs, just so you know right now, are what we're using. And one thing I didn't talk about is the validation data. CytSeq and TotalSeq is comparable to flow cytometry and CyTOF, and in some cases can be a little bit more improved. So very briefly, this is my last thing I'm gonna talk about here. This is my favorite example to really highlight the utility and perhaps even the future of TotalSeq and CytSeq technologies. This is a collaboration that we did with John Ashton from the University of Rochester, and he wanted to ask the following question. What is the transcriptional heterogeneity in leukemia patients? Well, that's a big question. It's a very hard question to ask. And before this technology, we wouldn't have been able to drill down in the way we have shown here. So very interesting question. And so what Dr. Ashton wants to do is he takes leukemia patients, isolates their bone marrow, and then does some proliferation assays at different stages of the disease. So here we have two patients, and then I'm going to show you what the transcriptional profile looks like in diagnosis and then in relapse. So first of all, both these patients are under the clinical diagnosis of leukemia, but yet we can look at the transcriptional profiles between patient 1 and 2. They're pretty different. Here in patient 1, we have some kind of spread out intensity here on the left quadrant, whereas in patient 2, we have a really centralized intensity in the lower right quadrant. And then if you look at the relapse samples, patient 1, there is a bit of a shift to a different kind of profile and archetype here, whereas in patient 2 at relapse, there's a totally different shift from intensity to the lower right quadrant to the upper left. And so if you can imagine, if you put together a patient from diagnosis, during treatment, post-treatment, relapse, you can really get a nice idea of the transcriptional effects with leukemia, for example. And so this is, again, a technology and a question that could not have been asked at this level. And again, because you're looking at the transcriptome, the RNA that is being made, and the protein simultaneously, this allows for unprecedented insights into what's going on in cells. Okay, so that's the last slide I have. Once again, thank you for your attention, everyone. My name is Angelo Moreno, and my email here is amoreno@biolegend.com. I know this is kind of a quick glance at everything that's going on at BioLegend. I am more than happy to follow up with webinars for your labs, or if you have any questions of technology, never hesitate to reach out. And lastly, biolegend.com, we have a lot of different tools that are available on our website. Every time I do a quick little webinar or meeting with somebody, if I do a website navigation, they're kind of amazed at all the different tools and technologies we have for flow cytometry and even all the different resources we have online. So always go take a look at that if you need some images or figures or even some flow cytometry assistance. And with that, I will take a break, and I think we might have a few minutes for some questions if anybody would like to ask any.
Thank you, Angelo. Thank you.
Yeah,
so
no
much.
problem.
A very clear overview of the last technology. So you can unmute yourself and ask the questions for Angelo. I will quickly ask, so how many, two things actually. So how many antibodies can you use for your total seq? So how many have been optimized, and do they include antibodies against intracellular proteins as well?
You asked like the two best questions, honestly. Yes, those are the most common questions. Thank you for that, Dr. Zuniga. So we don't know what the limit is. In-house, we have done up to 330 to date at once. Yeah, so the plexity is, we're just figuring it out. That sounds like a lot when you think about the max you can do for flow cytometry. But one of the important distinctions is instead of using the fluorochrome as its signature, you're using next generation sequencing. So you can really delve into, and the plexity is almost limitless, really. We're trying to figure that out. We have kits that are predefined panels available off catalog that have 10 markers up to 220. Yeah. And so as far as intracellular, that is, I'm glad you asked that, actually. We're looking into that right now. We're actually collaborating with John Cheng is helping us out, I think, a little bit with this to some extent too. So again, the collaborations are really important. And we're working on it. It's coming soon, hopefully, but that is the next big question. But as you can imagine, trying to measure something intracellularly, you want the oligonucleotide to be intact. It's a little bit of a different challenge, but we are working on that right now. We do have, one thing I didn't touch on is we have hashing antibodies and multiplexing antibodies for combining samples, and we have intranuclear hashing antibodies now. So we're bypassing the intracellular, we're going straight to the nucleus, so we're going to fill in that space in between very soon. Thank you.
Fantastic, thank you so much. So I think it's almost 11, and so we appreciate your time, Angelo, to update us, and I think that we should be closing. I don't see any questions in the chat, so I think that we should be closing the meeting. And again, thank you so much. Maybe you next